So last lecture is evolution of microbial life. I think all of you already remember what are microbes. Microbes means microorganisms. Usually when we think of microbes or microorganisms, we think of bad microbes, okay, things that harm us in some way. Remember that there are microorganisms residing in and on your body. Means if we could see with our eyes and we looked on our skin, you would see hundreds and thousands of bacteria and other microorganisms living on your skin. If you could go and check inside your any tract that has an opening to the outside, meaning your respiratory tract, your digestive tract, any other tract that has an opening to the outside, there is always microorganisms there. Some microorganisms that are coming in that you don't want, that are bad, but then a lot of the microorganisms, especially in your digestive tract, they are friendly, they are helpful, they help us, okay? So if you could take out all of those microorganisms that are present in and on your body, and if you could weigh them, they would weigh anything between 2 to 5 pounds. And human microbiota, this term is used for all of the microorganisms that are present inside or on our body. And we're going to learn about their evolution. So let's start with bacteria. If you take just a pin, this pin, and if you could enlarge it, and if you could see how many bacteria are there present on the point, on the tip of the pin, this is what the enlarged pin surface looks like. All the yellow things that you see are the bacteria, and the purple part is the pin. So now imagine if that small tip of the pin could have so many bacteria. Imagine you putting your hands on these desks, on these tables, you opening that door handle every time, how much microorganisms you're getting on your hands, or you're transferring to somebody else, right, to your friend. Now some of the most contaminated things you can find in your house is not the toilet. Some of the most bacteria containing stuff in your house is the sponge with which you clean your dishes, and the toothbrush with which you clean your teeth. You know that, right? So so the toothbrush that you use to clean your teeth, every time you put it back, it has so much bacteria, and that is why it is recommended that you change your toothbrush often. Actually, toothpaste is not even required for dental hygiene. That's something we use to feel fresh inside our mouth. What you really need is a clean toothbrush that you change frequently, like 3-4 times a year at least, minimum. We'll get a new toothbrush and then do it. Also, the way the restrooms are arranged, bathrooms are arranged, with the toilet bowl is there, and the toothbrush is open on the counter. Anytime you are using the toilet, you flush it, you know that aerosols are created, right? So the aerosols come and they could settle on your toothbrush. So toothbrushes have been found to contain fecal contamination, bacteria present in the feces that go and settle on the toothbrush, and then you use the same toothbrush to brush your teeth. And that's why this should be kept separate, or the toothbrush should be kept closed in a cabinet or covered the surface. Same for the sponges that you use to clean the dishes, because every time you're cleaning and it's always kind of wet. So this is an ideal environment for bacteria and other things to grow. So what do you do? Very simple. Some very simple things that you do is, at least every day when you use the sponge, after you're done cleaning, just put them in the microwave. Microwave them for 1 minute or 2 minutes so that it kills all. And then also regularly change the sponges, just like the toothbrush. Yes, Villa?
Wouldn't the soap kill any bacteria on the sponge?
Bacteria are very hardy. It won't kill all. And then as soon as because you've also put in water, and then when the concentration of soap is less and the water is more, and it stays there for a long time, you are getting a lot of bacteria again growing in that sponge. So then what do? We know the magic word, antibiotics, right? Antibiotics came and it changed the world. Penicillin was discovered. This was the first antibiotic that was discovered in the 1940s, and I might have described the story of how the first antibiotic was discovered. I don't know. So there was this particular scientist, Alexander Fleming. In those days, antibiotics were not discovered. He was working with fungal spores and other things, and he saw that once he forgot, in his lab he forgot the culture plates there, and when he came back, he realized that he was trying to grow some bacteria, and he saw wherever in his plates there was fungal contamination, there was fungal spores there, the bacteria had died. So he said, then that's when he realized he added 2 plus 2, and he said, so wherever there is this fungus growing, the bacteria died. Okay, there is something in these fungi that is killing the bacteria, and that's what led to the discovery of penicillin. So then he realized that penicillium, that was the name of the fungus from which he extracted penicillin. He extracted that and he treated the bacterial colonies with the fungal extract, with the penicillium, with the penicillin, and he saw that yes, that particular extract was killing the bacteria. So that was the first antibiotics, and since then this has worked like magic. However, because this has worked so well, what have we done as a community, as a society? We have overused the antibiotics, we have abused it. Why? How we have now tried, we have tried to use the antibiotics, apply them anywhere and everywhere, even when it's not needed. In the developing world, at least here in the US, doctors will not prescribe antibiotics as often, but in the developing world, antibiotics are so easily available over the counter. You just go to a pharmacy, you can get the antibiotics over the counter. And then people just take it by choice. As soon as they say coughing, day one, I'm coughing, that means I have some kind of problem. Let me just get an antibiotic. Why? Because it makes me feel good. As soon as I get the antibiotic, I do not feel sick and I am fine. But what has that led to? Remember the case where I explained how mosquitoes became resistant to DDT? Does anybody remember? How did that happen? What was the sequence of events? Yes, Sarah?
Well, it originally killed a majority of the mosquitoes, but then there was mosquitoes that had a gene that made them resistant to it, and then they bred, which made the population of that mutation bigger.
Right. So that means in a population there would always be some mosquitoes, that means there will always be some bacteria that have a resistant gene. Is it intentional? No. It's just by chance, random, right? Because mutations are happening all the time. So some bacteria, even if they don't have it at that time, at other point of time they might have a mutant gene which makes them resistant to that. So when we just take antibiotics, when it's not needed, we are killing most of the bacteria, which makes us feel good. But what have we done to our body? There were some bacteria that had the resistant gene. Now we have given them an open field to score, goals. So now they just reproduce and multiply, and they are the ones that become the majority of the population, and those bacteria become resistant to antibiotics, and then a community resistant develops. Meaning, let's say I did that. Let's say I have abused antibiotics, I took antibiotics, and now in my body there are bacteria that are resistant to, let's say, antibiotic X. Now I live in this house with my family members, I can contaminate, I can pass on those bacteria to my family members, right? So those family members, although they did not abuse the antibiotics, but they are now getting the antibiotic resistant bacteria from me. And then as a family, we could be spreading this to our neighbors, to our friends, to our relatives, right? And then as such, a lot of people in the community will get antibiotic resistant bacteria for no fault of theirs, just because 1 or 2 or 10 people in their community did that, made the mistake. So this is what happened and has happened, is happening across the world. If you talk to any nurse, any doctor that works in the ICU, you will hear that nowadays patients are coming in with pneumonia and none of the antibiotics are working. Okay, so pneumonia is a big killer for patients who are senior citizens, anybody who has respiratory troubles, sensitive. They all ultimately, AIDS patients, they all ultimately will die of pneumonia. So it's all an interplay between mutation and natural selection. Which finger was the natural selection? Yes. Which finger is mutation? Anybody remembers? Middle finger, yes. So mutation. So it's always mutation and natural selection. How some of the bacteria got the mutation, now they have a mutant gene, that mutant gene is resistant, and then natural selection is going to favor that. Why? Think of the natural selection from the perspective of the bacteria, not from the perspective of the human beings. The bacteria are also evolving, the bacteria are also mutating, right? So they are also subject to natural selection and mutation just the way we are. So for their survival and their reproduction, nature is going to favor the mutant gene that is helping the bacteria to survive. So mutations can be passed on from one generation to another as single gene mutations, or there have been cases which we call transposons. Transposons are jumping chromosomal segment where groups of resistant genes, they kind of stick together like close friends. So in a chromosome, let's say gene 1, 2, 3, 4, they're grouped together, they're all conferring resistance to a particular antibiotic, and these particular genes, they tend to jump together, okay? Now, a formidable superbug, this is the problem nowadays in the ICUs, it's called MRSA. It's called methicillin resistant Staphylococcus aureus. You see, this is written in italics. Why? Because it is a scientific name. So this is the genus, this is the species. This can cause fatal systemic infections. This particular bacteria is not getting killed by any of the antibiotics that we have now, okay? And systemic means whole body, so it can spread whole body infections. Often begins in settings where infection is common, such as athletic facilities, where you are, a lot of the athletes they are training together, they're sweating a lot, sweating always, warm and humid conditions are always good for bacteria and fungal infections, schools, military barracks, and so on. So this is how the MRSA superbug spreads. So our over-enthusiasm for antibiotics has actually caused the evolution of antibiotic resistant bacteria. Now this is just, we're talking about just the case of people taking antibiotics on their own. There are so many other ways in which antibiotics are being used. The livestock producers who are growing cattle and other things, they are, even if their cattle is not sick, they are using the antibiotics as a preventive mechanism, just like we take vaccines. Like, okay, just give them the antibiotics so that they don't fall sick, because in case they are sick, then I cannot sell this particular cattle, and then that's a financial loss. So to prevent financial loss, they give antibiotics to their cattle, to their livestock as a preventative measure, which is so bad because that gives rise to community resistance. Bacterial resistance against antibiotics. Sometimes doctors overprescribe antibiotics, and sometimes we make mistakes. Let's say a doctor has given you an antibiotic course to be taken for 5 days. You might have heard many people, or even you might have done it yourself. You take 2 days, medicine for 2 days, you feel better, then what do? Then you suddenly decide for yourself, because I'm already feeling better, I'm going to stop taking the antibiotic, because why over-medicate myself? Why take the medicine for 5 days if I'm already feeling better in 2 days? But what have you done? The doctor prescribed the medicine for 5 days for a reason. Why? Because that 5 days of medicine is required to kill the entire population of bacteria. But if you stop the antibiotic after taking 2 days, you might be feeling better, slightly better than before, or very much better than before. However, inside you are leaving some bacteria alive, and you are giving them a chance to become resistant to the antibiotic. Right? So if a doctor has prescribed you a given course, you are not supposed to stop it at your convenience. If you want to stop it, you need to talk to the doctor, ask them if I can stop that. Or whatever, 5 days, 6 days, 10 days, whatever is the course, you need to take it. This is what we covered. Let's see what's next there. Okay. Our invisible inhabitants, the microbiota inside, all the microorganisms that live inside our body, they they increase our susceptibility to infectious diseases, okay, if we disrupt that. If we change our microbiota, they can predispose us to certain cancers. They can contribute to conditions like this. So the good bacteria, the friendly bacteria that are present in our GI tract, that we need those bacteria, otherwise you won't be able to digest the complex carbohydrates that you eat. The complex carbohydrates that you eat, a lot of them, our body, human body, does not have the enzymes to digest them. So when those nutrients pass through your digestive tract, they go to the colon, and there it is those friendly bacteria that cause that digestion of those complex carbohydrates. They extract the nutrition, and that's how your body gets the nutrition. Now, when you take a medicine, anytime you take an antibiotic, it is killing not only the bad bacteria that gave you a disease, but it also, there's a collateral damage, right? So it's going to kill some of the good bacteria. Have you noticed, I mean, in some people, if you take antibiotics, you get diarrhea or some kind of GI upset? It's because the good bacteria are also dead. And what is surprising is I listened to a lecture by a very famous scientist who is a pioneer in the microbiota, okay, a field of science. And that scientist and his postdoc fellow, they did so many experiments together. They kept checking for the microbiota changes in their own feces and some volunteers over long periods of years, okay, and they saw every time you take an antibiotic and that antibiotic has killed the microbiota, the friendly bacteria, and then when you stop the antibiotics and the friendly bacteria start growing back again, the new colony of bacteria is very different from what you originally had. It is never the original exactly same group of bacteria or same kind of bacteria that you previously had. So the type of bacterial population that you have goes on changing like this based upon how many attacks of antibiotics and other chemicals you are putting your body you're putting inside your body, right? So the microbiota changes. Now why are we so interested in these microbiota? What's the problem if the bacteria inside our GI tracts change? Can anybody tell me what's your idea? Why are we so interested in talking about the microbiota inside our body? Yes.
You might have reactions to foods you never had reactions to before.
Exactly. Now the entire metabolism is different. Okay. Nowadays research focuses on metabolomics. You and I might be eating the same food, however our bodies might be reacting to the same food in different ways. Okay, we react differently to different foods depending on the enzymes, how they are acting in our body, depending on the microbiota in our body, how it is digesting. So I might be eating the same amount of protein, you might be eating the same amount of protein, my body might be extracting less protein from the nutrients, absorbing, more of it is just passing out unabsorbed, while you might be actually absorbing more of that protein. Right? So that is why with the same food, some people might be gaining weight, some people are not gaining weight, and different ways. Yes, Willow.
Is that how food allergies work?
Yes, food allergies can work because our microbiota has changed, right? So when the metabolism changes, our metabolites change. That means the way whatever we ate is being digested and absorbed is being changed. When that is being changed, that puts us at risk of diseases. That totally changes our physiology of digestion. So now some episodes in the history of life. Earth, what is the age of Earth is about 4.6 billion years ago. Okay, that's when Earth was formed. Now the two major groups of animals, all life forms are prokaryotes versus eukaryotes. Prokaryotes are very primitive kind of animals. Prokaryotes do not have a nucleus, they do not have a membrane, they do not have membrane-bound organelles, while eukaryotes, eu means new, they are more modern organisms. So prokaryotes came on, they evolved first, and actually they survived, for a long time before the eukaryotes came. So the prokaryotes lived alone for about 1.7 billion years before the eukaryotes evolved to give them company. Okay, so they have been on this earth for a longer time. That means bacteria has lived on the earth way longer than we have. So bacteria has an evolutionary advantage over us. And that is why, however hard we are trying, we cannot totally get rid of bacteria because they are smarter than us. They have lived, they have, survived on this earth for way longer. So their evolutionary mechanisms, their adaptations are way more advanced than us. They might be, we call them low, level organisms, but their adaptation mechanism, survival tolerance mechanisms is way more advanced than us. So this is some major episodes. If you want to get an analogy of how when prokaryotes came and when eukaryotes came. So if you start your journey here in Canada, that's origin of Earth. This is when prokaryotes came. So you've traveled from there to just to San Francisco, and the prokaryotes have come here. And then you look at eukaryotes came here, okay, that's Oklahoma City, and Homo sapiens there. So we are infants. If you think of evolutionary age, right, as compared to prokaryotes. Now eukaryotes, we evolved from prokaryotes. Prokaryotes were our ancestors. How? By chance. By chance, a mitochondria and a chloroplast were ingested, were taken in by a prokaryote. So the mitochondria got inside a bacteria and a chloroplast got inside a bacteria, and the bacteria did not digest it and eat it up. Instead, the bacteria or the prokaryote, whatever it was, it thought, oh, the mitochondria is actually helping me. It's a symbiotic, because the mitochondria started creating energy, generating energy for it, and the chloroplast also started absorbing the sunlight, okay, and started making food. So that living situation was beneficial, and that is how eukaryotic life started. The Cambrian explosion, we are talking about evolution in geological ages, which happened about 541 million years ago. This resulted or caused the evolution of all major animal body plants. Now at the end of Mesozoic era, which is 65 million years ago, more advanced flowering plants, birds and mammals, primates, they began to appear and they began to roam on the earth. The origin of Homo sapiens is only, forget about million years, it's 195,000 years ago, that's it. So when the earth was evolving, before life came about, the atmosphere on the earth was the conditions on the earth, life did not evolve for a long time because conditions were not ideal. Then slowly, because the earth was so hot, earth was super hot, okay, then as the earth started to cool down and the conditions around the earth, they also started changing. That is when slowly and slowly life started. But the question that scientists had been asking for a long time, how did life start? We understand evolution, okay, from simple organisms, multiple organisms might have evolved. But how did the first simple organism develop? Because the first life, if the life has to start, it has to come from non-living things, right? If we're talking about the first living thing, the first living thing came from all these non-living things. How did that happen? Is it really possible? Are we making a huge mistake, here? How did it start? So they started analyzing what the earth looked like, so 4 billion years ago. What was the surface of the earth? There was water vapor had already condensed and oceans were forming. Now what about the atmosphere? There was a lot of volcanic eruptions, and whenever there's volcanic eruptions, there are gases like carbon dioxide, methane, ammonia. These are toxic gases, okay? So these were the gases, these were getting released into the atmosphere. We don't have ammonia and methane nowadays in the atmosphere as much. We are polluting it, but not in the proportion that it was present at that time. So with these atmospheric conditions, how did life evolve? So there were experiments done, but before that there were hypotheses. So the hypothesis on the origin of life states that chemical evolution, this is chemical evolution because biological molecules evolved from chemical substances, okay? So there are 4 basic steps that you have to remember this hypothesis. The first hypothesis is the first organic molecule that formed, those were little things, monomers. Now those monomers, okay, such as amino acids and nucleotide, remember amino acids? Sounds familiar? Okay. What do amino acids make when they group together? Proteins. Good job. And what do nucleotides make? DNA, RNA. So these are the monomers. That means the first living molecule that formed was a very, very small molecule. These were monomers like these. The second step is, okay, the 4 stages, the second stage is these small molecules, the monomers, joined to form the polymers, the macromolecules, the big ones. So this has to happen. If this did not go into these sequence, life couldn't have evolved. So this is the second stage. What's the third stage? Now we have big organic macromolecules, meaning we got proteins made, probably, amino acids joined. We got nucleotides joined and made RNA or DNA. But where is the cell? The cell has not formed yet. There are only those organic macromolecules. So somehow these organic macromolecules, the protein or the DNA or the RNA that had formed, has to get inside a membrane covering. As soon as you cover something with a membrane, you have created the first cell, which is, we call it the pre-cell. right? It needs to have how do you define a cell? A cell has to have a cell membrane covering it. There has to be a definite boundary. Otherwise, there's no cell. So then that is what must have happened. And finally, that cell developed the ability to self-replicate. That means cell division, create baby cells. So that life could have continued. So in 1953, there was a trailblazing experiment done by the scientist Miller. It's called Miller and Urey experiment. And they, in their lab, they designed an apparatus that simulated, that means that copied the conditions that were present on Earth at that time. And that they kept that apparatus running for a week, okay? And then finally, they wanted to see if any organic, molecules had formed. So what they did, this is their apparatus. They had water here in a round bottom flask. They're heating it because the earth was still very hot, so the ocean water was bubbling, okay, temperature high, so water vapor being created. And what was there in the atmosphere in earth at that time? This is NH3 means ammonia, CH4 means methane, and H2 is hydrogen gas, okay. So they artificially supplied these gases into